Hi, Michelle Glass here. Welcome back to another episode of Chapter 10, Lecture 10. Our job here is to look at, well, what happens after that signal jumps from the neuron to the muscle fiber? How do we actually get muscle contraction to occur? So we're looking at what we can call the contraction cycle. Now, make sure as you're studying these processes that you know the details, every single detail. Okay, so go as specific and exact as you can in your memorization practice. Okay, so this is where we left off. So you remember this drawing, or maybe you drew your own, maybe you just wrote it out in words, maybe you're looking at the pictures, the artwork in your textbook. Remember, we had our action potential generated by the motor neuron, traveled the length of the motor neuron to the synaptic knob. When it gets to the synaptic knob, that triggered the opening of voltage-gated calcium channels. Calcium entered the synaptic knob, triggering exocytosis of acetylcholine. Acetylcholine diffuses out across the cleft and binds to our chemically gated sodium channels embedded in the motor end plate. Those channels open, sodium floods the cell. That is our action potential. So that action potential travels the whole length of the sarcolemma and then down into the cell at the T tubules. So it's where we left off. Our muscle fiber is depolarized. So what's going to happen next? Remember that structure called the triad. The triad was a sarcoplasmic reticulum, terminal cisternae, T-tubule, terminal cisternae on either side. Remember, inside the terminal cisternae, we have packed it full of calcium. When our action potential reaches the triad, it's going to open up voltage-gated calcium channels in the SR. So now calcium, which has been pumped into the SR, is going to rush out into the sarcoplasm. And this is happening throughout the entire cell. So if you have calcium released at one terminal cisternae, it is, well, terminal cisterna, then it's released at every single terminal cisternae. So it's happening across the board. When we talk about muscle contraction at the cellular level, we can say it's all or none. Either that cell is contracting or it's not. That calcium is going to go to your thin filament and it will bind to that troponin. The troponin, remember, is holding the tropomyosin over top of the active site. When calcium binds, it changes the shape of troponin, and the troponin then causes that whole complex to move away from the active sites. So now the active site is available, okay, where it wasn't before. So when calcium binds to troponin, the troponin-tropomyosin complex shifts off of the active site, and myosin head, who's been waiting and admiring that active site, it reaches down and it grabs hold of that active site, and we call this cross-bridge formation. When the myosin head grabs hold of the actin active site, we call this cross-bridge formation. We're going to see that myosin head already has been energized. It's been kind of revved up and holding on to this ATP molecule. Really, it's already broken down the ATP into an ADP and a phosphate, and it's been holding all of that energy. So our next step is to release that ATP and pivot that head, pulling that thin filament in towards the M line. So we can call this our power stroke, because you've released that energy and you're pivoting and pulling that thin filament in towards the M line. You're increasing your amount of overlap when you do this. So you're bringing your Z lines closer together. Oh, I popped ahead. Then we see a new ATP molecule comes in to bind. It is actually hydrolyzed by the myosin head, so that means it's broken down into an ADP and a phosphate. That energy is stored inside that myosin head. It's detaching and it's recocking, so it's described as energized. So that's always the starting point for that myosin head. So you've had cross-bridge formation, you've had power stroke, and then detachment. Okay, so that's one contraction cycle. Now, as long as your calcium is still available, you're going to do another round of cross-bridge formation, power stroke detachment, cross-bridge formation, power stroke detachment, until you're generating the amount of tension that you need in that muscle for the job that you need that muscle to do. Now, how do we return to normal? Well, remember the acetylcholinesterase is going to remove the acetylcholine that was in the synaptic cleft, so you end that signal coming from the nervous system. Calcium will get pumped back into the sarcoplasmic reticulum, and so that means it's coming off of the troponin. So we're going to see the troponin tropomyosin complex move back into place, with tropomyosin covering up those active sites. And then through other movements in the body or gravity or time, that muscle will gradually return to its resting length until the next contraction cycle. And that's it for now.